Kia ora koutou katoa, ko Mick Black toka wingwa, no mai, haere mai. Welcome along to our Friday afternoon Genomics Aotearoa seminar series. I'm Mick Black. I'm one of the co-directors of Genomics Aotearoa. I'll be hosting today's seminar. And today, as always, this is a seminar that I'm really excited about. It's actually a project that I don't know a lot about, except for what I've read in the project's annual reports. So I don't think I've heard you guys talk about the project today. So I'm really delighted to welcome along Björn Oback from the Bioeconomy Science Institute, Catherine Collins from University of Otago, and Ocean Mercier from Victoria University of Wellington. Fantastic to have you guys here. And we're going to hear from each of you about your aspects of the project. So I'm going to hand over to you guys and I'm going to get you to tell us all about tactical genetic control tools for rats.
Kia ora, ko Björn Oback tōku ingoa, no timano o ko tipona, e te noho o ki te whakatū. Tēnā koutou, tēnā koutou, tēnā koutou katoa. So thanks for joining us this Friday afternoon. It is indeed my pleasure to give an update of a piece of work that has been funded by Predator Free New Zealand, Predator Free 2050, for the past 3 years. And I'm hoping, Mick, that we will match what's in the annual reports when we go and talk about our different streams. For those of you who have been listening to Dan Tompkins' talk on genetic pest control a few months back, this will be a bit of déjà vu, but also an opportunity to fill in some of the technical details that he did not have time to go into during his seminar. So let's see if I can move this forward. Just a moment. Yeah, so as Mick said, it will be a joint session. There will be 3 research streams that we'll be covering. And the first one will be covered by me. That's about developing a transgenic suppression system for Norway rats. And then Catherine will talk about population genomics, and Ocean will talk about attitudes to genetic suppression systems with a focus on Mātauranga Māori. So it'll be pretty even. We'll sort of talk 15 minutes each. And I'll kick off. So research aim 1 of this project that's been running for about 3 years was to develop sex ratio distortion in Norway rats. And so just as a way of introduction, the only rats living in New Zealand are the ones listed on this slide, and they are all introduced. So in order of arrival, we have here the Kiore, or Pacific rat, or Polynesian rat, the Norway rat, also known as the brown or common rat, and then the black rat, also known as roof rat, ship rat, European rat. Our target for this project is the Norway rat or the brown rat, and I should say that the Norway rat is most likely not from Norway, but somewhere from Southeast Asia, but the name has stuck. So brown rats or Norway rats have established themselves in New Zealand in the late 1700s after visits from European sailing ships. And they are New Zealand's largest rats. They're good swimmers, but they're poor climbers, and they're abundant really only in towns and on farms and near water. They are largely absent from forests, particularly the southern beach forests where I am currently. So I'm in Nelson at the moment, and so we don't see a lot of them here. They eat seeds and fruit, and that's okay, but they also eat lizards and eggs and youngs of ground nesting birds. And as I only recently found out, they can also chew through walls and even through hard-shell suitcases. So they cause considerable damage, actually cause considerable damage to my bathroom and attic. So in short, they make a nuisance of themselves wherever they are, it's like humans. They're not easy to work with in containment, and so because they're feral animals. So we're using lab rats as a proxy. But that should be okay, because all lab rats, and there's sort of 28 strains of them shown here on this phylogenetic tree, are derived from the Norway rat. The lab strain that we use, the proxy that we use, is called Sprague-Dawley or SD rats. They are a specific outbred strain of the brown rat, except that they are not brown, but white. And to understand the approach that we are taking, this approach of sex ratio distortion, I'll basically take you back to understand sex ratio or sex determination in rats. As in all mammals, the sex of the rat is determined by the chromosome complement. So we've got XX and XY, XX for females and XY for the male. So there's a 50-50 chance as to whether an individual is male or female, standard Mendelian genetics. Now, on rare occasions, there can be examples of inheritance that is non-Mendelian or super-Mendelian. And you see an example of that normal inheritance on the left-hand side here, about a 50-50 if you follow the orange trait here. And on the right-hand side in green, we have a gene that is inherited in a super-Mendelian fashion, and those genes increase their chance of being passed off to the offspring. They are also known as selfish genes. So here is an example of a selfish supergene called the T complex in mouse. This is a naturally occurring gene, or rather a gene complex. In fact, that is a massive part of the mouse genome. It's about 1.5% of the mouse genome allocated to that supergene. It contains a range of so-called distorter genes, T complex distorters or TCDs, and a single responder gene. And I will come back to those responder distorter genes repeatedly during the talk. This supergene is protected from crossover recombination through multiple inversions. There's 4 inversions here, and they basically make sure that there's suppression of crossover during meiosis. And because of that, this gene complex has resisted mutations for the past 2 million years or so. So it's evolutionary, very robust. Now, the question is, how does this T complex actually work as a selfish gene? And so here we have a heterozygous male mouse, and you have in green the T complex, and in gray you've got the homolog chromosome without the T complex. And so when sperm are formed, sperm are haploid, each sperm will be either or. So it either can contain a wild-type copy here in gray, or it can contain a chromosomal copy with the T complex. The T haplotype can transmit itself at the expense of wild-type homologs by essentially affecting how sperm swim. And so when you look at this here, you can see those sperm that carry the T complex are able to fertilize eggs, whereas those that have the wild-type copy cannot. And this can lead to a sex ratio distortion of up to 99%, depending on a complex balance between these distorter and responder genes, and I will come to that. Now, rats do not have the T complex. They are naive to the T complex, and we predicted that if we were to insert the T complex on the rat Y chromosome, then one could produce sperm, Y-bearing sperm, that will be exclusively able to fertilize the eggs. And those sperm that carry the X chromosome would be not fertilizing. So in that situation, after a few generations of breeding, a population would contain more and more males and less females, and eventually there would be no females left, and the population can no longer reproduce and collapses. This is the strategy that we refer to as single-sex offspring selection, SSOS, or sex ratio distortion. This is not going to be 100%. As I said, the best case scenario would be 99%, but it could be less than that. And so we thought about including a sort of contingency mechanism here that would be to make the very few sneaker females that could go through sterile. And the way that we envisage this to happen is that those females will carry an autosomal gene, and this particular gene would be driving a Cas9-mediated knockout construct that knocks out a gene that is crucial for female fertility, and that gene is called NOBOX. So in that case, you would have sperm that would fertilize, and if there is a few females that are coming through, they would contain the NOBOX gene. And then again, by Mendelian transmission, half of the female offspring of such a transgenic male would be transgenic and become sterile. So these star females would compete with fertile females, reducing mating success and also using up resources. How does this work mechanistically? It's not fully worked out, but there's an analogy that people use, and that's sort of the poison antidote mechanism. Envisage that those distorters are the poison, so to speak, and the single responder gene acts as an antidote. And so in the spermatogonia, where the T cell, the T complex is present, they will be essentially immune against the action of the distorters. And so in this scenario, only sperm that carry the whole T complex distorter and responder will survive. As I said, don't take poison and antidote too literally. In the case of the T complex, the non-equal inheritance is not due to actually killing off the sperm. It is due to motility differences between sperm carrying the T complex and wild-type chromosomes. Now, it's really crucial for this whole concept to work that there is tight genetic linkage between the TCD, the distorters, and the antidote. So this T complex can only survive as a self-disseminating element or a gene drive when it is linked. And so the first step of this project was to break the linkage between the distorter and responder genes to study each gene separately. And that would also preempt any formation of a gene drive, because without the linkage between the two, a gene drive cannot work. So we started by designing constructs for autosomal integration and expression of a distorter gene. A distorter candidate that we used here is this gene called TIAM, which is a guanine nucleotide exchange factor that activates small GTPases, and another gene, the only known responder gene, called SMOC, which is a protein kinase. And both of those on their own, if expressed on their own, should actually impair sperm motility. And the second construct was a Cas9-driven no-box knockout transgene that should result in female sterility. This is what these plasmids look like, and there's a lot of detail on that slide, but I'm not going to go into all the details of the construct. The most important bit is that both contain testis-specific promoters, so that they will only be expressed in the male germline. Both contain chromatin insulators, so these are insulator sequences to shield the transgene from any positional effects due to random integration. And importantly, the responder SMOC gene has just been functionally validated in transgenic mice. The preprint just came out to show that this is a viable construct in an animal system. The no-box construct relies on Cas9 knockouts, and I'm not going too technical with this, just to sort of highlight, we designed guides against different exons of this no-box gene, 3 different guides targeting 3 different exons, and we used a system for the first time that allows multiplex gene editing. This was a system originally devised in rice, and we wanted to test this in a mammalian system. It relies essentially on making a polycystronic primary transcript. So you can see here, this is the primary transcript generated, and then every cell, every plant cell, and hopefully also every mammalian cell will have the endogenous tRNA processing system that will cleave that construct and release those 3 guides targeting the 3 exons, and that should boost the multiplex capability of that Cas9 system. So we made 2 plasmids, one with a constitutive CME promoter to test this construct in rat fibroblasts, and another one with a male germline-specific promoter that's only active in the germline. And this is what that looks like in rat fibroblasts. So these were transfected with plasmid with the triple guides, and you can see that if you compare the multi-guide RNA with the single guide RNA, that for every location here, those 3 different editing sites within no-box, the multi-guide RNA construct is at least as efficient, if not more sufficient, than the single one. But importantly, the effect here is additional, so it's cumulative. All these 3 add up, and so that means that you effectively triple the chances of getting a knockout. So editing efficiency with such a multiplex construct is essentially 100%, 98%, as opposed to single constructs, which were about 30%. After validating those constructs in vitro and one of them in mice, we then decided to go into rats. And so this is the process of trying to generate transgenic rat lines. You collect fertilized oocytes, zygotes, and these are then injected into the pronucleus with those different constructs, these different germline competent constructs, hopefully integrate there randomly, as I said initially. Then those zygotes are transferred into gestational surrogates and then grow, hopefully establish a pregnancy, and then the pups are being looked at to check whether the founder pups are transgenic. And this is a summary balance sheet for the pronuclear injections that were undertaken. And you can see that about, close to 1,200 zygotes were injected, roughly equal numbers for those three different constructs, SMOC, which is the responder, TM, which is the distorter, and NOBOX, which is the knockout gene. About half of them survived. Then 562 embryos were transferred, 36 pups were born, 30 of them were genotyped, and unfortunately, the number of transgenic founders so far is 0. So what we can observe from that is there's a very low survival of the injected zygotes and very poor in vivo development of the transferred embryos. Only about 6% of embryos transferred actually resulted in viable pups and no transgenic founders at this stage. So to conclude that first research stream, we have designed a sex ratio distortion strategy. We've constructed and functionally validated transgenes for germline-specific expression in mouse for those two transgenes. The NOBOX knockout construct was validated in vitro in rat fibroblasts. They were all injected into rat zygotes to generate transgenic rat lines, and an average of about 10 viable pups per transgene was obtained, but none of them carried the transgene. And that concludes sort of the first phase of this project as we envisage this, which is really about capability building, developing foundational concepts and transgenes for rat suppression and containment, and hopefully has set up this project for potentially extending into the actual generation of founder rats from existing transgenes, breeding and phenotyping in a potential next stage. We'll see how that goes. So just to acknowledge the people that have worked on that work stream, the team at Ag Research, Götz Leibel, Susie, and Si Ming were the two postdocs working on the project, T-complex and the NOBOX project, with support from Bridget and Sally, our collaborators at the Max Planck in Berlin, who actually discovered the T-complex in the first time and described it in the late 90s. The pronuclear injection was undertaken at the University of Auckland, and training for the staff, so Siming and Susie, was done by Fabian at the Macquarie University in Sydney. And with that, I will stop sharing and pass the baton on to Catherine Collins.
Ko Catherine Collins toko ingoa no kaitawhahau kei Otipoti toko kāinga. My name is Catherine. I'm based down in Dunedin at the University of Otago. And I'm going to be talking to you about the second aim of this project. So this was focusing on population genomics. So I was carrying out this work alongside Professor Martin Widenwerther, who is currently overseas at the moment, so it'll be nighttime for her, but she's based at Plant and Food Research and also has a position at the University of Auckland. And Dr. Brooke Whitelaw was working as a postdoc down here at Otago and did the actual hard part of this in terms of handling all the data and that kind of thing and all the analyses. So our aim was to develop genomic data sets that allowed us to look at gene flow in both the Norway rat and the ship rat. And the idea behind this was to get an understanding about, once a gene drive system was developed, how might we go about introducing that into natural populations, and what do we know about movement between populations of rats within Aotearoa. And then have a look at other considerations for genetic control, specifically looking at some of the variation, for example, present in some of the regions that Björn's team were working with. Just a little bit on the history of the introduction of rats into Aotearoa, because this could inform population structure, or kind of, I guess, kind of has an implication in terms of the of population structure. Like Björn said, the Kiori or Rattus excellens was the first rat to be brought over when people first arrived from Polynesia. And then with the arrival of Europeans came the arrival of the Norway rat in the 1700s, and then not too long after Rattus. Once Rattus arrived, it seemed to largely outcompete the Norway rat in many parts of the country. And of course, both of them also outcompeted the Kiori as well. When we began this project, our understanding of population structure in both of these species was based on around about 500 bases of the mitochondrial control region, and this was some work done by James Russell a few years ago. And so, can you see my cursor there? Cool. Sorry, I should have checked that beforehand. And so James and his team, they identified that there were two main haplo groups of the Rattus norvegicus, and that was kind of one mainly in the North Island and one mainly in the South Island. There was a wee bit more diversity in the mitochondrial control region of norvegicus. We can see kind of areas such as Auckland where we've got higher levels of diversity, possibly due to the kind of higher connectivity between different populations. And one kind of predominantly in the North Island, also represented down here in the southwest of the South Island. And I'll come back to that later on. For this particular project, with the number of individuals that we were able to sequence, we were looking kind of a broad picture approach, looking at kind of what was the bigger picture in terms of the population structure across Aotearoa, as opposed to zooming in into some of the finer detail. So we had a little under 100 samples for Rattus and just over 70 for Rattus norvegicus. And we had a pretty good distribution over the country there for Rattus. More missing in norvegicus, but Björn has already spoken to some of the differences in the distribution of those two species. The tissue that we were working with was collected over a period of around about 20 years, and this was largely, again, thanks to James Russell, and a lot of the samples that we were working on were ones that he had looked at the mitochondrial control region a few years ago. With them being a pest species, most of it is opportunistic trapping from trapping groups. And once we had Brooke on board, she did a lot of work talking to different trapping groups as well to bump up that sample size, which puts us in a really nice position for perhaps zooming into some of those finer scale questions going forward that some of this data set has arisen. And we aimed to have at least about 8 times coverage for all of our samples, and we also selected a few from different locations to have some higher coverage sampling as well. First off, while the whole genome was mapping, we had a quick look at the mitochondrial genomes, and this really just confirmed what James's team had identified previously. We had that kind of higher diversity in regions such as Auckland, and we could see kind of a hint at some level of population structure when we look across the country. When we first looked at the whole genome data, our first apologies here for some of my text is covered. The first, I guess, attempt at this, we followed kind of standard variant calling protocols, and we used GATK to call SNPs, filtered out our data set. So we had a really kind of high quality data set. This left us with about 60,000 SNPs, but we were quite bamboozled when we saw some of the results, and we just couldn't see any hint of population structure there compared to the mitochondrial genomes. Alongside this, we were also attempting to merge this data set with some other New Zealand data sets. And unfortunately, Brooke was running into issues with a batch effect with these samples, and it just could not be resolved in the end. As part of that, though, she was exploring other ways to have a look at the data sets and ultimately landed on looking at the genotype likelihood estimations, which we have found, like others have found, to be quite useful for this particular context, looking at the population structure. And so when looking at this data set, it made a wee bit more sense kind of intuitively based on the distribution and the biology of the species, as well as that earlier control region data. Generally, we can see this kind of like an isolation by distance pattern, this kind of climb from northern North Island when the darker red down to southern North Island and the lighter red, and in the orange in Rattus. And then again in the South Island, we've got this kind of spread from northern South Island, sorry, and the lighter coloured blues and greens up here, and then the darker coloured blues and greens southern South Island. And this kind of matches up to that mitochondrial haplogroup that James had seen that was present on the north of the North Island and the southwest corner of the South Island as well. In some regions, we do have kind of a few more samples where we can zoom in a little bit more detail, and we can see some kind of have a look at some of the nuances of these regions and think about how might the introduction of gene-drive rats work in these kind of places. So this here is that cluster that you could see on the kind of upper part of the PCA of my previous PCA. It's not just Aotearoa, but some of the surrounding islands around Great Barrier as well. So you can see kind of some of the island populations do look to be relatively isolated, and perhaps could be showing these clusters due to kind of bottleneck effects and founder effect and that kind of thing. Whereas other parts of the North Island, we've got kind of more genetically similar samples spread out across a wider area. So it'd be quite a nuanced kind of method that you might use for the introduction of these rats. Moving on to Rattus norvegicus, the distribution of the sampling, like I said, wasn't so great as the Rattus. We see that kind of that predominant North Island haplogroup up here, and then there's a couple of distinct South Island haplogroups down here. So again, that kind of aligns with that control region work that James had done earlier. Looking at the genome-wide data, there was a little bit more comparative data from international literature for Rattus norvegicus compared to Rattus, and that probably makes sense with the interest of the norvegicus being used in the lab as well. The New Zealand rats were distinct from those from their kind of native range in China, and also from other global populations, so the United States, parts of Europe and Southeast Asia. When we zoom into those New Zealand samples, again, we can kind of see that we've got this, this is that main, probably representing that main same, I guess, population as that main mitochondrial haplogroup that we saw in the North Island of the, of Rattus norvegicus, and those handful of South Island samples kind of here in the middle. So I think we still have kind of more work to do in terms of sampling across the South Island to get a better understanding there. And again, we can zoom in the parts of the North Island where we've got higher density sampling, and we can see that we've got that similar kind of climb, if you like, of that kind of identity. The more closely geographically samples are to one another, the more close genetically they are to one another, and as we move further away, genetically distinct. One of the other things that we were interested in with this data set was to kind of link the data back to the work that Björn's team was doing. And so Björn introduced you to the SMOC gene, which was one of the groups in the T haplotype that his team were working with. This was definitely one of those areas where it raised more questions that are answered when we began to look at the SMOC genes in Rattus and Rattus norvegicus. We could see that variation in this T haplotype was quite complex. Here we've got a phylogeny of different rodentia, mitochondrial genomes in a phylogeny. And then we've got the the presence of the different SMOC genes in the Rattus norvegicus and also in Rattus rattus. So you can compare, we've got, I jotted these numbers down here. We've got, if I think of it's about 13 SMOC genes in the Rattus norvegicus, compared to 9 in the mouse. So it's there's been a number of different SMOC genes that we've got that some have been duplicated, and we've got some present in the rattus that are not present in the mouse. And so there's still some work to be done in terms of understanding what is the function of all of these different SMOCs, and redundancy and the presence of them and that kind of thing as well. And you can see here in the synteny plot too as well, the number of inversions and duplication events and that kind of thing that have occurred between the two different rat species as well that we're looking at. So there's still a lot more work to be done in terms of kind of unteasing what this might mean in terms of the implications of a gene drive. So with that, I will just acknowledge that a number of different researchers across the country have contributed to conversations around this, and as well as a number of trapping groups as well have contributed samples. And I will mute myself, stop sharing my screen, and pass the rākau onto Ocean.
O tēnā koutou katoa, ngā mihi ki a koutou o Genomics Aotearoa mō tēnei wānanga, kia koutou ko tai mai ki te whakarongo kia mātou. Ko Ocean Mercier tōku ingoa no Ngāti Porou, and I'm here in Te Whanganui Ātara. So, yeah, very happy to present on our work on focusing on kaitiaki perspectives on SSOS for rat control. The title here is a nod or to the broad range of work we did, and this I want to really acknowledge that we as a research team came through the bioheritage pathway onto this Predator-Free 2050 project, and through our work on perspectives in the development of gene drive and other genetic technologies for the control of invertebrates, particularly pest wasps and later varroa mite. So want to acknowledge the wonderful team who's worked across both projects, Bioheritage and Predator-Free 2050, my co-lead Sarah Belcher, research assistants Alan King-Hunt and Rain Hananui, and guests from the USA who spent a summer with us charting perspectives of conservationists and environmentalists on gene technologies for pest wasp and pest rat control. So the team has entrusted me to share a little bit about our work, but I cannot cover everything in the time that we have today. So folks, thank you for your trust and advance forgiveness for any missing bits or misrepresented bits. So our key goals were to identify tangata whenua concerns about the impact of rats on taonga species, but also interests, opportunities and concerns they have regarding single-sex offspring selection type options for controlling pest rat populations. And in there I've got to sort of who was over which parts of the project, and in grey I've presented work that is really important to the project generally, but that I don't have time to cover today. So what I will present to you today is a mixture of mine, Rain and Alan's work on those perspectives, and particularly Alan made a call to focus in on perspectives, tangata perspectives with people who identify as kaitiaki, and that drove the criteria for recruitment and selection of interviewees. And also note that Rain's work covering 100 plus interactions through surveys and interviews across Bioheritage and Predator-Free 2050 projects was a really important piece of work that draws out the sorts of questions that the people we were talking to about these technologies were asking. So I want to acknowledge Phil Lester here, who led the Tranche 1 and Tranche 2 invertebrate pest control using novel technologies work, a co-author on this particular paper, and thanks, Dan, for the shout out a few weeks ago on some of our work here. This is co-led this paper by Natalie Jones, who herself is a tangata whenua pest wasp volunteer control person, and draws upon her master's research supported by Bioheritage, along with some of the WPI student work on conservationists and environmental specialists' responses and ideas about gene technologies. And also in this paper is a version of this graph here. So I couldn't let my colleagues present graphs without me presenting one. So I'll stay on this one for a little bit. Some of you may kind of recognize variations of this published in Amanda Black's paper a few years ago, which harks back to the big survey, as I call it, that was commissioned by Edie McDonald and her team through Bioheritage, which solicited more than, I think it's around 9,000 total responses and a significant number of Māori responses, over 1,000. And it was interesting to go back and look at this data to see what it told us and what directions it might set for our work in speaking one-on-one with kaitiaki participants. So on the left, there's bars and there's a line graph here. The line graph, it's the y-axis on the right there, and on the left is a Likert scale that refers to the bars. So people were asked about their general attitude to 9 different pest control methods, 4 of them traditional known ones that included hunting and trapping, and most people settled in around a sort of comfortable and will accept with appropriate controls on a scale between 1, should never be used, and 5, no concerns at all. They showed less comfort with poison by air type approaches, with sort of some of the cohorts that we zoomed in on, particularly the Māori cohort, averaging around 2, that should only be used as a last resort. By contrast, some of the newer technologies, people felt more or less uncomfortable with. For some of them still uncomfortable, particularly for Māori respondents here, but would accept with appropriate controls. So one of the other things this graph tells us is the increasing level of uncertainty as participants went through the options, the 9 options, one by one. And so you can see the uptick in percentage who are responding, not 1 to 5, but 6, I don't know. I don't know what I think about gene drive, Trojan female. The uptick is most pronounced at the end there for those particular technologies, with quite a large number of Māori, general population Māori of 42% actually opting out of presenting, or not opting out, but saying that they don't know about Trojan female, what their attitude is there. So, there's an increased uncertainty, I guess, here, which we just need to keep in mind when it comes to how we engage people in these kinds of conversations, and how important it is to present some form of explanation. That particular survey had hoverovers with short descriptions. And actually, if I go back to that, you'll see that some of the descriptions at the bottom, for instance, mostly male offspring, infertile males, say a little bit, even in a short amount of time, short number of words, about the effect, as opposed to a label, gene drive, Trojan female, the effect of the gene technology. So this is a screen grab from the primer that Susie Tsakas had worked on as something that would support Alan's work in speaking with kaitiaki. And this was it was a few minutes, about 6, I think, video that each of our kaitiaki that we, and I say we because I did actually do a couple of the interviews, but Alan did the lion's share, But what this was important to communicate to the people that we spoke to, that obviously this technology needs to be safe, needs to be effective, but that it also needs to be guided by public opinion and supported by scientific policy. So just to give you a little bit of a snapshot of where the people that we interviewed fell in terms of their positions on SSOS and pest control. There was a group that one of our collaborators from here at Te Kawa Māui, who we brought in, suggested could be framed as kia mai, a group that saw the issue that if we're going to get to a predator-free 2050 NZ, then using new techniques like single-sex offspring selection could be a crucial step towards actually achieving that. So with strong agreement there, double thumbs up.
Well, for time's sake, maybe I'll just sort of settle on these two slides as representing some of the actual kōrero we got. So on this, the Kia Maya group were, talked about how we need complementary tools, not just one, if we're going to actually commit to and achieve a predator-free 2050. Some people were really amazed by the technology, so friggin' amazing, and it's got to be a new innovation if we're actually going to achieve this big goal, with another participant actually calling it a taonga, what the team had developed here. Another participant talked about how we need something that is at landscape scale to achieve not just eradication on islands, but across the mainland. The other group, the Kia Tūpato, Be Careful, and the Kia Matara groups, talked about how the power of nature would sort of intervene, and that it might work for a time, but then sort of nature would fight back, as it were. So this group tended to be quite skeptical and wary of the technology, and ask questions about how are we as humans are controlling our own behaviours, looking at ourselves as creating problems rather than looking at the rats. One person talked about how triggering it was for them to think about editing fertility, and felt triggered to think about the sterilisation and the history of what's been done in that space to Indigenous peoples around the world. And another noting that this is not from our mātauranga, this is a sort of a way of doing that's been imported from Western science. So quite illuminating is some of the questions that they're asking across the board, but focusing in on the single-sex offspring questions that are coming out, because some of these questions we can answer with some confidence, actually, such as the one about could this cause a worldwide extinction of Norway rats if they escape overseas, through to questions that we're working on in this project, such as, how long would it take for SSOS to work through the modelling. So a really insightful group of questions here that not only, that give us a sort of a direction to go towards in terms of shaping our research to public perceptions. Okay, just a bit of a summary, and I think for time's sake, although I've got other things to share with you, maybe I'd stop after this one. Just wanted to, again, draw on what Dan said the other day about empowering informed decision-making. And just this morning, through my Google Scholar alert, Sylvia Nissen's paper came through, and the question that she's asking about who, for what purpose is this engagement for, if it tends to be sort of one-directional or one-sided. So it is important that we engage and work together as much as possible to work through some of these thorny issues that are being raised by kaitiaki and by the public more generally. So, but one thing that is critically important is that in any of these interactions, we need information communicated well. And we, let's see, that questions about GM and the ways that questions are being asked tell us something of people's priorities and can inform our development of science and communication. And we need safe and engaging ways to promote discussion on gene tech in contexts that include, that are, as Sarah Hartley says, broad and inclusive, that give space for people to air their historical, ethical, cultural, social, etc., considerations and concerns. This is some work that we're doing through Bioheritage on an engagement activity. Actually, this is a game prototype that we've developed that enables people to talk through their, they work towards a scenario in which you achieve a healthy environment by addressing long-term scenarios while meeting short-term challenges through a different suite of methods, and you do it collaboratively, so you either all win or all lose. And yeah, that's a lot of words. So I'll just maybe end on my final slides, which is just a little plug for some of our publications and a bit of an insight into what we're working on to finish off this project and future publications that are a bit of a watch the space. Also, because I get all the Google Scholar alerts for the Dearden et al. Paper all the time, but actually, if you like that paper, you might also like this one here, led by Simon Palmer, published a few years ago in Journal of Royal Society of New Zealand. Ka pai, kia ora.
I will take this opportunity to thank you all again. Thank you very much for a wonderful seminar. And I will thank our audience for coming along on a Friday afternoon. I hope the weather is nice and sunny where you are, just as it is in Dunedin. It's not often I can say that. We'll be back again in 2 weeks. We have got Ariana Lord from Harvard Museum of Comparative Zoology, I believe, speaking to us in a couple of weeks. So that's going to be a really exciting one. So thank you again to our speakers. Thank you to all of you for coming along, and have a fantastic weekend. See you next time.